So we're going to go over the steps for a DNA extraction. First, we want to be able to break up the cells in order to release the DNA. You can think of a sample we get from the wild. All the DNA is locked inside its cells, so we need to lyse those cells in some way in order to release all that DNA. Once the DNA is released, we can bind that DNA to something solid. We usually use a filter. And so once the DNA is bound to that filter, we can wash away everything else that's not DNA. That could be protein, that could be bits of cell membrane. Anything that's not DNA gets washed away so that all we have on our single filter is just DNA. Finally, once we isolated the DNA, we can release the DNA from the filter by changing, again, the chemical components of the solution that will release the DNA, and then we can collect that volume, which contains all of our DNA, that will eventually send off to sequencing. So we've isolated our sample, which in this case is a small zooplankton that we collected from a net tow. That was out on Catalina Island, but you could also do this for any sort of sample that you collected that's microscopic or macroscopic. And so what we'll do is we'll take we'll take our sample and we'll pipette it into our tube here. And to this tube is where we'll do any of the chemical or physical lysing we needed to do to break apart the cells in order to release the DNA that we'll eventually sequence. And so this tube will go on into the further steps. Our sample came back from any of the lysing, and we can begin the next step of binding the DNA to a filter. We need to bind the DNA to a filter in order to hold on to the DNA so we can get rid of everything else. So how we do that is by pipetting a certain amount of the sample onto a filter tube or a spin column. Now this is extremely cool. This white part at the very bottom is an actual filter that will bind the DNA under certain chemical conditions. And so we have a number of solutions that we can use to enhance those chemical conditions. But first step is we need to get our sample into our spin column. This is a micropipette. So this is a micropipette. It's essentially a very fancy turkey baster where it allows you to suck up a very precise amount of liquid in order to pipette it into somewhere else. So how it works is there's a plunger at the top here, and this will push down to push all the air out and will be released to suck up the sample. You'll also push this back down to expel any of the sample that comes in the tube, but you can also set the volume here by changing this little dial. And so this is how you can get very precise measurements of volumes using this dial, set it to what you want, and then go ahead and suck up your sample. Now we use certain tips for these. Now this is a larger volume pipette. It can pick up 100 to 1 milliliter, or 100 microliters to 1 milliliter. And we have certain pipette tips that allow us to pick that up. And so if you were to put one on, you can see how the tip goes over the pipette and it allows us to suck up a sample. And then eventually this disposable pipette tip we can shoot off and allow us to get another one, so to avoid any cross-contamination amongst our samples. And so that is a micropipette. And so we're going to use this micropipette to suck up some of our sample. As you can see, it is now in the tip there. And we're going to pipette that into our spin column. And hopefully you'll be able to see our spin column increase in volume. The sample we have there. And one of my favorite things to do with the pipette is you get to shoot the tips out when you're done with them, because they're all disposable, at least the tips are. So now I have added my sample to the tube. This includes our DNA as well as some buffer. And you can see all of our liquid is in the top portion of the spin filter with the filter at the bottom, and there's no liquid in the bottom of our tube just yet. the next step, we'll spin this tube and pull all of the liquid through the filter, across the filter to collect into the bottom. So we're able to spin these small tubes with a centrifuge or a microcentrifuge. Here you can see it has little openings for each of the tubes to fit into. And so we're going to place our tube that we just pipetted all of our liquid into one of these slots. Now, one especially important thing to know about centrifuge is that because it's spinning very, very fast, you need to make sure that they're balanced. And so in order to balance that, we'll put another tube that doesn't have anything in it, we call it our blank, on the other side that is the same weight, just so that the centrifuge doesn't kind of go out of control when it starts spinning really, really fast. And so we can close this, and as soon as I close it, it's going to start spinning. So at this moment, we have all of our liquid has passed through. Remaining on the spin column should be just our DNA. And in the bottom, what has passed through is any of the contents of the cells that were lysed before, any of the extra buffer that was sitting in there in the initial solution before we spun it through the spin column. So we want to keep priority on this spin filter because that is where all of our DNA is that we want to isolate. So at this point, we've added a little bit of an elution buffer to the top of our spin column. It's a little hard to see because it's a lot smaller volume. And what this is going to do is allow the DNA to be released from the spin the filter and be collected into the bottom of a tube. Now, I've taken this spin filter out because I'm going to replace the tube that it goes in with a separate tube now that it's clean. That will be our final tube that contains all of our DNA in it. And so I placed my spin filter into this tube, and now it's ready to be centrifuged again. Again, we want to balance this with something of equal weight, and so I've taken our blank and put it into another tube that also has the cap kind of dangling off the side in order to help balance our centrifuge out a little bit. Again, when I close it, it'll start up. Work that time. Great, and so we can pop it back up. And if we pull out our sample tube, again, it's going to be a smaller volume, but you can see a very small amount of liquid at the bottom of the tube. It's about 20 microliters. And just to give you some reference, we started with a sample size of about 600 microliters. And that is about half a milliliter is 500 microliters. So we've really taken our large sample and condensed it into this really small volume, which should be fairly concentrated with the DNA that we extracted. This becomes a very precious tube that you will eventually put on ice while you wait for the rest of your samples to be extracted. And then this is what gets sent to the sequencer. So at this point, you've extracted all of your DNA sequences through the process we just described, and you have sent them off for sequencing. Sequencing can be fairly complicated, but there is a very nice video that I've put a link to in the Blackboard content folder for this lab that you should watch to understand how Sanger sequencing works. And so take the time to go watch that one video right now, and then return back to this video. Now, what we'll do next is show you how all this can come together. And so from Sanger sequencing, you see that it produces a plot of literally colors that you can read off as different bases. And so let me show you how that can be interpreted. And essentially, these hills that are in different colors, and each color corresponds to one of the four bases. A's are green, C's are blue, T's are red, and G's are black. Now you can see there's generally only one peak at each of these base positions. And so at this position here, it's a green peak, so we call it an A. And similarly, we have a next A. And then it switches, so the green drops all the way down because it was not registered at that time point, but there was a lot of blue. So you see a blue peak, and that corresponds to C's. And so if you stick all this together, you'll essentially get this DNA sequence, G, A, C, T, T, and so forth, all the way down the line. And so what I've done is I've taken 4 DNA extracted samples, and I put them in this one aligned FASTA file. And so this should look somewhat similar to you, all the A, T, G's, and C's here. The reasons why they have these dashes and a bunch of the ends, and dashes even in the middle, is because what we had to do is, this is the same gene that we sequenced for every individual. So these 4 individuals here, these are 4 samples. They need to be aligned. They're sort of the same gene that has a certain length. And so the sequences must be that same exact length. And so up here in the file, we have all sequences aligned, just to show you that this grouping here, this sequence here, is the same length as this sequence here, and the same length as this sequence here. That's what alignment means. And that is just needed so we can compare these 4 sequences together to see how related these individuals are. I've also went ahead and annotated these. And so for 3 of our 4 organisms, I blasted the sequence, and then I came up with the annotation for it. So this is the organism that the sequence aligns with, and it is a type of copepod. And so I've done that for 3, but we have one mystery sequence down here, which I want you to blast. Now, all you have to do for this is you can take the entire thing like we have been doing, copy and paste that, and put in that into blast. And what you can do there is, once you find out that organism, this is important, you need to delete just the question mark here and leave that arrow right at the beginning. That arrow tells our the downstream processes that this is a new sequence. And so in front of this, or right after this arrow, is where you'll write the name of the organism. Unknown sequence name is what we'll call it. So you want to make sure this is on its own line with no spaces in there, similar to how we have above here. So we're calling it unknown sequence name now, but you will provide the actual organism's name. So what we'll do is, now that we've changed this, you can save this. We'll save as, just to show you need to make sure it's saved as a .fasta. Save that somewhere you know you can find it again. So once you have this file saved, you need to go to this site here. It's essentially a tool that will take sequences that are aligned and annotated and put them into a phylogenetic tree. A phylogenetic tree essentially shows us the relatedness between different organisms. It could be organisms of the same species or of different species or different phyla. It essentially looks at the same region of the sequence, so that aligned sequence we have there, and can assess how related those are based on the number of differences between all of their bases. So for us, we only have 4 individuals, if you remember this, 1, 2, 3, 4, and the last one is the one that you will name. In order to input this is fairly simple. You're going to choose File. And then for me, I'm already in the location, but you need to find this aligned underscore, allseq underscore aligned underscore annotated .fasta file. And you just say open. Now, there's different methods that can generate a phylogenetic tree. For us, let's use the UPGMA method. This is going to make our tree look a little nicer. And so that is all set. Everything else can be default. And then all you need to do is say submit. And so it's not going to take all too long. And then we will have our phylogenetic tree. So you can see we have some copepods grouped together, and then a decapod grouped even more closely with that grouping. And then our unknown sequences is sort of the least related to the rest of these. So how you read a phylogenetic tree is things that are closer together as far as their branches go, they are more similarly related. So these 2 copepods are closely related, the most closely related among these groups. And that makes sense, they're both copepods. And so so this is a group here, right, of 2 copepods. If we backtrack out, the next sequence that we would come upon is this decapod sequence. So this decapod is more closely related to these 2 copepods than this unknown sequence is to these 2 copepods. However, these 2 copepods are more closely related to themselves, to each other, than to the decapod or the unknown sequence. And so this is one group here of, crustaceans, we'll say, this, arthropods. And then we have this outgroup, which is this unknown sequence that you will find out. And it should make sense once you figure out what that is. And so the goal for today's lab is to produce this phylogenetic tree with the appropriate name of the organism here. So the assignment will be taking a screenshot of this tree, putting that into a Word document, and giving a 2 to 4 sentence description of the tree, and tell me if it makes sense or not, the tree as it falls. Given that once you find out what this organism is here, does it make sense that this organism is the outgroup to the rest of these? One issue that you may run into is if you choose a file that is not a .fasta. So say I just choose something that's Coral text like that, and I change this again. It should say error in reading input sequence. Please check your input. Now, it's most likely due to you saved it as a .text file and not a .fasta. So that is one issue. Another issue is if when you're renaming this unknown sequence, if you happen to accidentally delete this arrow in front, it will also not be able to read it in. So you need to make sure you have that arrow in the very front. If you need any sort of reminder, just look at the ones above. The ones above are in the format that works.